Hi everyone, welcome back to my channel. And today we are going to cover our chapter Microbes in Human Welfare. Okay, and it is chapter number 8 of Biology class 12th. Okay, we till now we have completed our two chapters. First one is Principle of Inheritance and Variation, and second one is Evolution. Now third is today Microbes in Human Welfare. It is a easy chapter, and not very tough. Yeah, and it's not much lengthy. It's just medium type. Okay, so today I have taken like easy chapter because we have already completed our two tough chapters. So today I thought that we can take an easy chapter, and because we have to cover NCRT of all of these three chapters we have to read. We have to practice PYQs of all three chapters. So for that I have taken today this easy chapter so that I can give time to PYQs and NCRT reading also. And let me tell you, today I'm going to give a test of all these three chapters after this. Okay, and I'll show you sheet tomorrow. Okay, practice sheet on which I'm going to write. Okay, and this will be I think 40 to 50 marks or 20 marks, three chapters. Okay, I'll tell you how it will be. Okay, and so let's start our chapter Microbes in Human Welfare. So microbes, what are microbes? Microbes are tiny organisms. Okay, these are harmful also, and these are helpful also in human welfare. Means in human, how they are useful in humans, or how they are like harmful to human. Okay, helpful and harmful. Okay, so let us read. Several microbes such as bacteria, virus, fungi. See, now in this fungi, some calls it fungi and some call it fungi. Okay, in class 7th where I was teaching with he was like science teacher, and he was calling it fungi, and he have like said to me it's not fungi, it's fungi. Then I started speaking it like fungi. And when I became in like 8th class, there is a chapter of microbes, second chapter. There I called fungi. Then my ma'am said that it's fungi beta. Yeah, so pronunciation doesn't matter. You should just like focus on the topic. Yeah, we can like call it fungi. And yeah, if someone say it's fungi, call fungi. If someone says it's fungi, so call fungi. And if someone says it's fungi, so say fungi. My if I tell about me, so I'm calling it fungi. Okay, so fungi, etc., are useful to human being in many ways. So how they are useful? They are useful in many ways. Which microbes? Bacteria, viruses, and fungi. Okay, now microbes in household products. Microbes in household products, that the products which we are using in our household, means in our daily life, how in that they are helpful. Who? Microbes. So yeah, sorry for disturbance. Okay, as I told you, the washroom is attached, so I have to open the door for uh, my family members. Okay, so let's start. We should focus on this. So microbes in household products. First is lactobacillus or lactic acid bacteria. LAB. Okay, like we have read in earlier classes also. 8th class I have read. I remembered that lactobacillus and lactic acid are like somewhat related to curd and milk. Yeah, so yeah, it converts milk to curd by producing acids that coagulate means um, something like freeze something, not in that way, but yeah, something like coagulate, convert, like process, and like liquid to a semi liquid. Yeah, so that semi liquid and solid. So it converts milk to curd by producing acid that coagulate and partially digest the milk protein Okay, so what lactobacillus does, bacteria does, it is also called lab, lactic acid bacteria. Okay, so it converts milk to curd, okay, by producing acid. Okay, which acid? Lactic acid. Okay, that coagulate and partially digest the milk protein. Okay, now a small amount of curd added, how this is done. So when we add a small amount of curd to milk, okay, okay, to milk, then as inoculum is or starter are same, inoculum and starter are same. Like when we add a small amount of curd or like liquid form of liquid form of curd, like when curd is there, when we just coagulate it, so we get what? Oh, that liquid form of curd. So if we add that liquid form or curd to milk, any container in form of inoculum or starter, then it is converted into curd, fully curd. Okay, so full milk is converted into that, and this starter contain million of lab. Okay, at suitable temperature, multiply and convert milk to curd at specific temperature. Okay, they convert and multiply and convert milk to curd. It also increases vitamin B12 in curd. That is why, like, I don't like drinking milk. Okay, so I like eating curd. So yeah, that is also like advantage ki, see, curd also contains very good amount of B12 vitamin, and milk does not. Okay, so yeah, in stomach, lab helps to check pathogen. Okay, check pathogens. Fine. So this was about microbes in household products in as a lactobacillus. Okay, bacteria. Fine, what we have read, it converts milk to curd. Okay, how? When we add inoculum or starter. Okay, also B12 vitamin. Okay, it multiply at specific temperature and convert. Okay, and one more thing was the stomach, and lab is used to check the pathogens in our stomach. Okay, now second is baker's yeast. Baker's yeast. So baker's yeast is also known as Saccharomyces cerevisiae. Saccharomyces cerevisiae. See, they are like pronunciation is okay. If we have read this about 2 to 3 times, then we are able to learn it. But their spellings are like very difficult. Okay, S A Sec SACC Cro CHRO myces MYCES. Okay, cerevisiae CEREVISIAE cerevisiae. Okay, so it is used to make bread. Okay, by fermenting dough, it is used to make bread. Baker's yeast. Okay, now bacterial fermentation in like anaerobic respiration form in dough is used to make food such as dosa, idli. Okay, so bacterial fermentation is used in making the like anaerobic respiration, in which anaerobic respiration occur, and so is used to making like dosa, idli we are making. Okay, the puffed up appearance of dough is due to production of CO2. See, w- like we have taken any dough of like corn flour. Okay, and we have, when we'll put and we'll just dough it, and when we'll do separate, we separate and we put in oven or micro wave. So what we observe, we observe that their size is increased and they are puffed up. Why? Because there is CO2 evolution. Okay, so when CO2 evolve, the puffed up appearance is because of CO2. Okay, toddy is made by fermenting sap from pulp. Okay, so toddy is like a bread in south. Okay, it is made by fermenting sap. What is sap? It is produced from palm leaves. Okay. Okay, now microbes are used to ferment, okay, fish, soybean and bamboo shoot. Microbes are also used in fermenting fish, soybean and bamboo shoot. Okay, what we have read in baker's yeast, we have read that it is used in making bread. Okay. Okay, used to make bread by fermenting dough and bacterial fermentation as anaerobic respiration, in which the we use, like, we make the food, dosa, idli, these type of foods. And the puffed up appearance of the dough is due to the evolution of like evolution of CO2. And toddy is a like South Indian bread type structure that is made from sap, and sap is produced, fermenting sap, and this sap is made from uh, palm leaves. Okay, and microbes are used to ferment fish, soybean and bamboo shoot. Now next is cheese. Now how cheese are made? So cheese are made, is a oldest food item produced by microbes. Okay, means from a long time cheese is like made from these microbes. Okay, Swiss cheese. Now it's new. Yeah, Swiss cheese. Like when we were watching Tom and Jerry, so we used like Swiss cheese. So what is between difference between cheese and Swiss cheese? So cheese is basically evenly distributed all over, but Swiss cheese is where there is like hollow circles on its surface. It's unevenly. Okay, not flat basically. Okay, so that is like large holes due to production of CO2 by Propionibacterium shermani. Okay, so because in Swiss cheese, why the that hollow circles were there? Because of large CO2 evolution. CO2 is evolved in all processes, but in Swiss cheese that was like large holes due to production of large CO2. Okay, by Propioni Bacterium shermani. So we have read three, like Lactobacillus, Saccharomyces cerevisiae, and one more, Propionibacterium shermani. Okay, now and one more, there are three types of cheese, and one is Roquefort cheese. Roquefort cheese ripened by growing a fungus. It is ripened from a fungus. Okay, on that. Now, microbes in industrial product. We have seen how microbes are helpful in household products. Now we are going to read about industrial products. Okay, so production of beverages. Okay, like drinks, okay, antibiotics, etc., on industrial scale require growing microbes in very large vessels. Okay, ferment, and these large vessels in which all this process is going on which these microbes are growing, that is known as fermenter. Okay, now first is fermented beverages. Okay, how they are useful in, microbes are useful in fermented beverages, let's say. So, Saccharomyces cerevisiae, which was used in brewer's yeast, baker's yeast, okay, is used in the production of beverages by fermenting malted cereals and fruit juices to produce ethanol. So, when we in this basically process we use beverage brewer's yeast to make beverages, and that is Saccharomyces cerevisiae. Okay, this is used for like fermenting fruit juice or malted cereals to produce ethanol. Okay, okay. Depend on the type of the raw material and type of processing. Okay, urine and oh, what I said, urine. Oh, sorry, wine and beer produced without distillation. So There are two process followed by fermented beverages. One is distilled and second is undistilled. Okay, so undistilled one is wine and beer. Okay, so wine and beer produced without distillation, and whiskey, brandy, rum produced by distillation of formed broth. Okay, now second industry can be of antibiotics also, like medicines. So antibiotics, chemical substance produced by some microbes. So some microbes are responsible for like producing chemical substances. Okay, and some are used for like killing or retarding the growth of pathogen. What are now pathogen? So pathogens are like disease causing microbes which cause diseases. Okay, these are also a kind of microbes only, microorganism. Okay, so in antibiotics some like microbes are useful, like responsible for like producing chemicals. Okay, these chemicals are like useful in killing or like killing the disease or retarding the growth of like pathogen in our body. Okay, first is penicillin. So what is penicillin? So penicillin is first antibiotic. Okay, penicillin is first antibiotic. Okay, and it was discovered by Alexander Fleming. Okay, by Alexander Fleming. So what he observed is he observed that streptococcus, streptococcus, see, lactobacillus, uh, Saccharomyces, Saccharomyces cerevisiae, uh, Propionium, Bacterium, Shermani, and fourth one is this Staphylococci could not grow around a mold. If there is a mold, so it cannot grow. Who? This antibiotic, this pathogen. Okay, he observed that Staphylococcus could not grow around a mold. Okay, penicillin, Penicillium notatum. Now, fifth name is Penicillium notatum. Okay, so he observed that a fungi named Staphylococci could not grow around a mold of Penicillium notatum. So he extracted extracted penicillin from it. So from penicillin notatum, he just extracted penicillin. Extracted means take out it. Okay, so he just extracted penicillin from it. Okay, now two scientists, sorry, came and they what they established, and who were the scientists? So they were Ernest Chain and Howard Florey. Ernest Chain and Howard Florey established its full potential as an effective antibiotic. 19 okay. So this penicillin was established and given the full potential as an effective antibiotic, and these both scientists who Ernest Chen and Howard Florey have prized by Nobel Prize. Okay, in 1945. Okay, and okay, this where this penicillin was used, so it was used in like treating American soldiers in World War II. Okay, they are used to treat plague, whooping cough, diphtheria, leprosy. Okay, plague, okay, whooping cough, okay, diphtheria and leprosy. Okay, now third is how they are useful in bioactive molecule, how they are useful in enzymes, how they are useful in chemicals. Okay, so first is coming is our organic acids, like chemicals. Okay, so in sorry so in aspergillus niger, a fungus. It is a fungus. Okay, what Aspergillus niger, it produces citric acid. Okay. Second is acetobacter acid. It produces acetic acid. Okay, because as a name acetobacter. Okay. Now second is Clostridium. Clostridium butylium. Okay, Clostridium butylium. Okay, this is a bacterium. Like these all three are bacterium which I am going to. Lactobacillus lactic acid, a bacteria. Clostridium butylism, bacteria, butyric acid. Acetobacter acetic, it is a bacteria and it produces acetic acid. Okay, who was producing citric acid? So citric acid was produced by Aspergillus niger. Acetobacterium, acetobacteria, acetobacter acetic bacteria produce acetic acid. Okay, second was butyric acid. And yeah, butyric acid. So butylic S. Clostridium butylium. It is used for producing butyric acid. And lactobacillus is used for bacterium lactic acid. And second is lactobacillus. Okay. Now, second is how they are used in making uh, alcohols. So, in alcohols, Saccharomyces cerevisiae, a yeast, is used for like producing ethanol. Okay, and how they are used in producing enzymes. So, first enzyme is lipase. Okay, so these microbes are useful in that also. Used in detergent formulation. Okay, detergent formulation. Okay, and these also remove oily stains from the laundry. So like lipase, I was like, is used in our body also. uh, Maybe I'm wrong. Lipase or ligase, I just don't remember, sorry for that. But yeah, we should remember that lipase are used in the laundry for removing oily stains and for like formulation of detergents. Second is proteinase and protease. Okay, no, pectinase pectinase and protease to classify bottled juices. Okay, to clarify means to clear, to clean those bottled juices means bottled fruit juices. Okay. Streptokinase. What is streptokinase? So these are produced by streptococcus. Streptococcus. Okay, streptokinase is produced by I think it's a fungi. Yeah. Oh, streptococcus. Used as clot buster. Okay, these are used as clot buster to remove clot from blood vessels. Okay, so let me tell you, from this topic it is a matched type question can be formed. Like they can give all five like enzymes and they can give their functions and they can just say for matching. Okay, and I have seen this question, I have done. So to like remove clot blood vessels, clot buster. Okay, and patient who have myocardial infarction, they are also treated with streptokinesis. Fourth is cyclosporin A. Cyclosporin A, produced by Trichoderma polysporum. Okay, and it is a fungus used as an immunosuppressive agent in organ transplantation. So basically, cyclosporin A is used in patients who are like who wanted organ transplant. Okay, and these are used as immunosuppressive agent. Okay, and it is produced by Trichoderma polysporum. It is a fungus. Statins. What are statins? So it's an enzyme which is produced by purpureus. Monascus purpureus, and it is a yeast. So used as blood cholesterol lowering agent. Okay, it is used to like suppress or lower down the cholesterol level in the body. Okay, and it also inhibit the enzyme responsible for synthesis of cholesterol. Yeah, it is like lowering agent of cholesterol, so it will also inhibit the yeah. Our next topic is microbes in sewage treatment. Okay, so sewage or municipal waste water contain human excreta and other organic waste. Okay, it also contains a number of pathogenic microbes. Pathogenic means which like contains pathogens. Okay, so should not be passed into rivers, streams, and water bodies. Yeah, if they contain pathogen or pathogenic microbes, should not go in like water bodies, rivers. Okay, yeah, in streams. Yeah, we know that. So it is made less polluting by passing it through sewage treatment plants. Okay, so like we cannot like stop it, but we can reduce its like waste. Like we can reduce the number of pathogens. Okay, or we can just kill them. So it is made less polluting by passing it through their STPs. STPs are sewage treatment plants. Okay, so it include two stages. Okay, they include two stages. First is primary treatment and second is secondary treatment. So let us read about primary treatment. So what does primary treatment says? It is the physical removal of particle, like physically with like if any big containers are there, so physically like with hands we can remove those. Okay, removal of floating debris. Floating debris means the waste which is floating on the water bodies. Okay, by sequential filtration, by like first with hand then filtration we can do. Yeah, so removal of what we remove? The removal of great uh, soil and pebbles. See, first we have done physical collection of that floating debris. Then what we have done? Then we have done filtration. Okay, then we have done sedimentation of what? Soil and pebbles. Soil and pebbles. Pebbles are like small stones, like yeah, which are like dissolvable or which is not dissolve cannot be dissolved in like water. So we can remove that by sedimentation. Okay, the settled solid from the primary sludge which in which primary treatment is going on. Okay, sludge, the supernatant form of primary effluent. Okay, from primary sludge is a form of. Okay, so the that settled solid, when we remove that and we pass to that primary sludge, and in primary sludge that is a primary effluent left. Okay, supernatant form of primary effluent. Okay, then we go to secondary treatment, that is also known as biological treatment. Okay, primary effluent is passed into large aeration which is left from like primary sludge, that is now primary effluent. Okay, is passed into large aeration tank and constantly agitated means rotated. Okay, and aeration means in which air is revolving. So primary effluent is passed through a large aeration tank and constantly agitated. Okay, this allow vigorous growth of useful aerobic microbes. Okay, useful microbes are there, their growth they like allows. Okay, aerobic microbes into flocks. What is flocks? Bacteria associated with fungal filament to form mesh-like structures. Okay, what are flocks? The bacteria associated with the fungal filament to form like mesh-like structure. Okay, these microbes consumes the organic material in the effluent. So what does this helpful like microbes does to us? So they what they does is they consume all the organic material that is present in the effluent, primary effluent. Okay, this reduces what it does now by like reducing the organic compound. So it does like this reduces the BOD. What is BOD? biological oxygen demand. So oxygen demand is reduced. Why? Because organic matter is removed of the effluent. Okay. BOD, amount of O2 consumed by bacteria to or oxidize all organic matter in 1 liter of water. So for oxidizing the organic matter in 1 liter is like for oxidizing bacteria needs amount of water. Amount, say what is BOD? Amount of O2 consumed by bacteria to oxidize all organic matter present in effluent. Okay, and if there is like we have first uh, just removed or killed, I think those microbes consume organic compounds. Okay, if they consumed organic matter, then if organic matter is only not left, then means their BOD is also reduced. Yeah, means there is no there is no uh, matter left to oxidize, to whom bacteria will oxidize. Yeah, so like there is less amount of BOD. So it measure, or it is the measure of organic matter present in the water. Yeah, okay. The greater the BOD, is it polluting, and greater it polluting potential. So if BOD is higher, then polluting potential is also higher. Okay, if that is only less, then means organic compound is not there and the water is less polluting. Okay, the effluent is then passed into a settling tank. Okay, where the bacterial flocks are sedimented. So now the when BOD is reduced, when microbes are not those microbes are like bacteria, and no work organic compound is reduced. Yeah, then that effluent is passed to settled tank. Okay, and there those bacteria are left, those bacterial flocks are left here, they are sedimented. Okay, so this sediment is called activated sludge. Now this settled tank is called activated sludge. Okay, the material, the liquid present in that settled tank is called as activated sludge. Okay, a small part of activated sludge is pumped back, okay, into the aeration tank. This small part is added next to the first one, primary, secondary one, first part. Okay, that was aeration tank to serve as inoculum. Inoculum is a starter. Okay, the remaining sludge is pumped into large tank. Okay, where it is known as anaerobic sludge digesters. Now anaerobic, something is left, substance are left, so this sludge will digest that. Some anaerobic bacteria digest the bacteria and fungi. So okay, some anaerobic digest the bacteria and fungi in the sludge by producing gases like CH4, H2S, CO2, biogas. These are biogas mainly. Okay, the effluent is released into natural water bodies. Now this effluent is released in natural water bodies like rivers and streams. Okay, now the Ministry of Environment and Forest has initiated Ganga Action Plan and Yamuna Action Plan to save from water pollution. Okay, yeah. So this was sewage water treatment. Now, we have read how microbes are useful in household product, how microbes are useful in industrial products, and we have also read how microbes are like useful in like treating the sewage. Yeah, and now we are going to see that how microbes are used in producing the biogas. Okay, so let's start. Biogas is the mixture of gases. Okay, okay, containing dominantly methane. It contains like in dominating amount in like Dominant and recessive we have read. So in dominant amount it contains methane, CH4. Okay. Produce, and this is produced by microbial activity and uses fuel. Biogas is produced by microbial activity. Okay. As we are studying microbes chapter, so of course this is like produced by microbial activity, and it is also used as fuel. Okay. Certain bacteria which grow anaerobically, okay, or cellulosic material, okay, produce large amount of methane, okay, along with CO2 and H2. Okay. Now, are methanogen and common bacteria, methanobacterium. Okay. So, certain anaerobically like bacteria or cellulosic material produce large amount of methane, CH4, along with H2S, along with CO2 and H2, and methanogens, and some bacteria, particularly bacteria, some common bacteria, methanobacterium. Okay. They also found in anaerobic sludge during sewage treatment, and during sewage treatment also these were found. Okay. These bacteria are also present in the rumen. What is rumen? A part of stomach in cattles. Okay. So these bacteria, okay, rumen, what they does, they break down the cellulose and provide nutrition to cattle. So the cattle which eating the like grasses and these plants which contain cellulose, high amount of cellulose. So what these microbes does, they break down those cellulose and gives the cattle the nutrients from that cellulose. Okay. The excreta means dung of cattle, okay, which we say is like gobar. Okay. Rich in this bacteria. Okay, these they are rich in this bacteria. Okay. Dung can be used to generate biogas. Okay. That is also known as a gobar gas. So what is now biogas plant which generates like biogas? Yeah, so let us see. Biogas plant consists of a concrete tank, okay, 10 to 15 feet deep, okay. Bio waste are collected. Okay, bio waste means like biology waste, like fruits, the dungs and the mm, yeah, you know, bio waste. So a slurry of dung is fed. Okay, a floating cover placed over the slurry. Okay, which keep on raising as the gas is produced by slurry. Okay. The biogas plant has an outlet which is connected to a pipe, okay, to supply biogas to nearby house. Okay. So there's a biogas plant has an outlet which is connected to a pipe, and to supply the, why it is connected, to supply the biogas to near households, nearby house. Okay. The spent slurry, the slurry which is now spent, which is unused, which is like used, and it is not, now it is not usable, now biogas is not generated from it. Okay. That is removed, okay, through outlet, and can be used as fertilizers. Now this can be used as a soil or fertilizers in soil, like manure types. Okay. Now biogas is used as fuel for what? For heating, for cooking, for lighting. Okay. Now, it is eco-friendly and pollution free source of energy. Okay, these are the like, we can say like advantages of biogas. Why should we use like biogas plants, or why biogas is such an important gas? Okay, so it is used as fuel for like heating, okay, cooking, lighting. Okay, it is eco-friendly and pollution free source of energy. Okay. Technology of biogas was developed in India by whom? So Indian Agriculture Research Institute, Indian Agriculture Research Institute, IARI. Okay. And Khadi and Village Industrial Commission. So these two questions were also asked. Okay, organization's name which does biogas technology developed like these two. Okay, now our next topic is microbes as biocontrol agent. We have read household, industrial, sewage treatment, biogas, producing biogas. Okay, now next is microbes as biocontrol agent. How they control this life, how they control this biology. Okay, bio means life, yeah. So biocontrol is the use of biological method for controlling plant disease and pest. So in like plant disease also there, not only about humans and animals, but it is also used in plants. Okay, like and pest. Okay, chemical pesticides and insecticides. Okay, kill both useful and harmful organisms and cause pollution. So what they says is the pesticides or the insecticides we bring from the market, chemical, they contain many chemicals, and they even they kills the harmful microbes, but they like kill the helpful microbes as well, so that they cause pollution only. Yeah, so they are talking about that only. Example also they have given lady bird beetle. So beetle with red and black making feeds on aphids. Okay. Dragonflies prey upon mosquitoes. Okay. Like these black with red and black making feed on aphids. So they are like killing one species. But that species is like contributing to the like that food chain also, no? So it's like okay, that is useful, that it is like responsible for like mm, causing disease to a plant, but it also cause, it does not cause, like it is also helpful for the food chain. Yeah, and for many ways, in many ways. Okay, so dragonflies prey upon mosquito. Okay. To control butterfly caterpillar, the dried spores of Bacillus thuringiensis (BT) are mixed with water. See, sprayed onto vulnerable plants such as bassicus and this fruit trees. Okay, these are eaten by caterpillars. Okay, in their gut the toxin is released that the larva get killed. Okay, the scientists have introduced b bacillus thuringiensis (bt) toxin genes in plants. Example, bt cotton. From plant bt cotton they have introduced bacillus thuringiensis. Okay, fourth is now dragonflies upon mosquito. Okay, to control butterfly caterpillars. Okay, bt cottons, ladybird beetles. Okay, fourth is trichoderm species, also free living fungi common in root ecosystem. So effective biocontrol agent of several plant pathogen are like using this. Okay, baculovirus genus is it's a genus where it's like nucleopolyhedrovirus, and this attack insects and other arthropods. Okay, these are suitable for species specified narrow spectrum and insecticidal uh, epi- application. Okay, they have no negative impact, okay, on plants, mammals, birds, fish, and non-target insects. But this is desirable in IPMC, integrated pest management. Okay, who baculovirus. Okay, program to conserve beneficial insects. Okay, so this was like theoretical. We should learn it. Okay, yeah, there were five baculovirus, trichoderm species. So many questions are coming from third part of this microbes as biocontrol agent, that is BT cotton. Okay, so BT cotton is like is a plant from which the scientists have introduced B. bacillus thuringiensis. Okay, and these are like eaten by caterpillars, and in duct the toxin is released, and the larva which is like there, that is killed. so that no other population came of there. Now our last second topic, that is, yeah, last topic only, that is microbes as biofertilizers. So biofertilizers are organisms that enrich and nutrient quality of the soil. Example, bacteria, fungi, cyanobacteria. Okay. Rhizobium, symbiotic bacteria. Okay. Root nodules of leguminous plants. Okay. It also fixes atmospheric N2 in organic form. So many questions are coming from this part. Yeah, I have seen many. For, in every sample paper, in every PYQ, rhizobium, mycorrhiza, or cyanobacteria, or full like 5 marks, microbes as biofertilizers is coming. So yeah, this is also important. Free living bacteria, example of azospirillum and azobacter, enrich the nitrogen content of the soil. Okay, and they can fix atmospheric nitrogen. Yeah. And now what is used in like mycorrhiza? So mycorrhiza is like symbiotic association of fungi with plants. Okay. Many members of genus Glomus, okay, from mycorrhiza, okay, absorb phosphorus from the soil and pass it to plant. Okay, they like absorb. They are also like root something, and they are also contributed to root rhizobium, but they are root nodules of leguminous plant. But these are like absorb phosphorus from soil and pass it to plant, give resistance to root borne pathogen. Okay, they stop the root-borne pathogen. Okay, tolerance to salinity and drought. Salinity means salty area and drought area. Okay, they have like tolerance of that. They can tolerate that atmosphere. Give overall increase in the plant growth and development. Okay, and now a third one is cyanobacteria. So this is like blue-green algae, a type of blue-green algae, and this is autotrophic microbes. Okay, autotrophic microbes, it's not heterotrophic. Yeah, I remembered my 8th class from this autotrophic and heterotrophic. But see, these are autotrophic microbes. Okay, they make their own food, like they does not depend on anybody. Okay, they can fix atmosphere, and for example, anabina, nostoc, and oscillatoria. It adds organic matter to the soil and increase its fertility. So if they are used as biofertilizers, so it's like a basic point that they will increase the fertility of soil as well. Okay, so this was all for today. This was chapter microbes in human welfare. Okay, this was easy chapter, that's why this is covered very like easily, and approximately in I think 1 hour it's completed, or before 1 hour. So yeah, we'll just, what I'll do today is, I have covered 3 chapters. I'll revise all three from starting. Which topic I'm facing, like this is difficult, I'll note that, I'll paste on my table. Okay, and I'll revise all days. Like I'll revise all topics like from up to, but I'll read those topics very carefully and very deeply. Yeah, I'll revise, and maybe today, now, or like tomorrow morning, I'm going to if I'm going to solve all 3 chapters test. I'll say to ChatGPT to provide me a test of all these 3 chapters, and I'll give the test in a way that I'm giving my board paper. Timer will be the water bottle, pen, and sheets I have. Okay, so I'll get to know the attemptation procedure, what is going to be. So yeah, PYQs also, NCRT also, but now I'm not going to do PYQs, no NCRT. Firstly, I will go to revise these all topics. Yeah, so hope you liked this session, and thank you for watching. Stay tuned.